Cellular respiration is a process that all living things use to convert glucose into energy. Glucose is the primary molecule of energy production through the cellular respiration. However, other carbohydrates, lipids, and even proteins can be used by the body alternatively. These molecules are broken down and enter cellular respiration at various stages. Cellular respiration is a collection of three unique metabolic pathways, which are glycolysis, Kreb cycle, and electron transport chain.
Glycolysis is the first step in both anaerobic and aerobic respiration, which occurs in the cytoplasm of the cell. This process does not require any oxygen, which is why it is common for both types of respiration. Glycolysis starts with a molecule of glucose, which has 6 carbons. This glucose molecule will be broken down into pieces during glycolysis. In the first part, fructose 1,6-bisphosphate will be formed. Fructose 1,6-bisphosphate is a very unstable compound. This process requires 2 ATP. Next, fructose 1,6-bisphosphate splits into two types of 3-carbon molecule, dihydroxyacetone phosphate DHAP and glyceraldehyde 3-phosphate G3P. Eventually, all DHAP will be converted into G3P. After that, G3P will be converted into pyruvate. This process will produce 4 ATP and also 2 NADH. Pyruvate will then be modified into 2-acetyl coenzyme A and also 2 molecules of carbon dioxide using 2 coenzyme A. This process will generate 2 NADH. To sum up, at the end of glycolysis, 4 ATP will be generated, but 2 ATP will be used up to start up the process. 4 NADH will also be generated. These 2-acetyl coenzyme A molecules will be used in Kreb cycle later.
Hi, my name is Kueh Chen Wah. My matrix number is 70118. Today I'm going to discuss a topic about Kreb cycle, or known as citric acid cycle or tricarboxylic TCA cycle. This Kreb cycle is used for aerobic organisms to produce energy through a series of chemical processes by oxidation of acetate. Usually, the Kreb cycle takes place in the matrix of mitochondria. Right now, I will going to introduce how this cycle works. Kreb cycle will be total with 8 steps in this cycle. Firstly, the step 1 starts from citrate. Citrate comes from the glucose we intake. The glucose we intake will undergo oxidation of pyruvate and become acetyl-CoA. In the first step, 4-carbon oxaloacetate will combine with acetyl-CoA, undergo citrate synthase to become 6-carbon citrate. In the second step, citrate will become 6-carbon isocitrate by undergo aconitase by removal of water and addition of water. In the third step, 6-carbon isocitrate will become alpha-ketoglutarate by undergo isocitrate dehydrogenase process. This is because a reduction is occur in this process. NAD+ will become NADH and 1 carbon dioxide molecule. That's why 6-carbon isocitrate become 5-carbon. In the fourth step, 5-carbon alpha-glutarate will become 4-carbon succinyl-CoA. This step is almost alike to the step before this. This step is known as alpha-ketoglutarate dehydrogenase. In the fifth step, 4-carbon succinyl-CoA will become 4-carbon succinate by undergo succinyl-CoA synthase. 1 ATP or GDP with phosphate group will become ATP or GTP produced. In the sixth step, 4-carbon succinate will become 4-carbon fumarate by undergo succinate dehydrogenase from FAD to become FADH. FADH is then transfer its electron directly into the electron transport chain. After that, in the seventh step, a water molecule is added into fumarate to become malate. This process is known as fumarase. Lastly is also the eighth step. The eighth step is also the last step of citric acid cycle. The 4-carbon malate will become 4-carbon oxaloacetate by reducing NAD plus to NADH and 1 hydrogen molecule. This process is known as malate dehydrogenase. Therefore, oxaloacetate can be reused again for the next acetyl-CoA. And this process is keep repeating to change glucose and form energy reuse. Therefore, we can notice that in this Kreb cycle, for a singleton of a cycle, 2 carbons will enter to the acetyl-CoA and 2 carbon dioxide are released. Then, 3 molecules of NADH and 1 molecule of FADH2 will be generated, and lastly, 1 molecule of ATP or GTP is being produced. That's all for the Kreb cycle. Thank you.
Electron transport chain is the last stage of aerobic respiration. This process occurs in the mitochondria of animal cells. To be specific, it is occur in the inner mitochondrial membrane, which is arranged into folds called as cristae that increase the surface area available for the transport chain. To undergo electron transport chain, some reactants needed are FADH2, NADH, GTP, and carbon dioxide. And the end product of this process are NAD+, H2O, and ATP.